Hey, so this video is something I wanted to do in class, but my internet died. So I wasn't able to talk with you, all of you that are doing this project during class. I just wanted to show you a little bit, give you some guidance about how to figure out what 23andMe does. Like, what are their methods? How do they determine something like ancestry? And we won't go into all the details because there's a lot. Explain how 23andMe determines which genetic markers indicate a particular ancestry. A lot of the human genome is the same from person to person. Like every single one of us has the same set of DNA. There's only a few particular places on the genome that vary between people. So those are the genetic markers they look at, and they look at, so are there certain genetic markers, certain variants that are found in certain areas of the world, and then they say, well, that's indicative of an ancestry in Finland or wherever. So I'm not going to expect you to be able to analyze everything that I'm going to go over right now. I'm going to just point out a few things that are interesting and something for you to think about, something that you could discuss in your project. Picking just a couple of them will probably be sufficient. I'm, again, just going to give you a lot more than you'd have time to really explain adequately. So you kind of pick the highlights that you think are interesting. But keeping a couple things in mind when you are looking at their methods, when you pick a couple things that I point out to you and you expand on them and explain them, think about this question of are are these methods, or is analyzing DNA truly reflective of somebody's ancestry and their identity? What does this imply generally for, human society, as well as for that specific person that's, telling you about their report? What does this idea that we could sort people by DNA really imply? How did we even come to the idea that there are races at all? And then, of course, coming along with that, where did racism come from? What do DNA tests do? Do they reinforce the idea that there are races, and do they reinforce racism, or do they do the opposite? There aren't any clear boundaries between races, that everyone's mixed race. And then does that then decrease the racism that we see? Why or why not? So you really could go either way there. That's your thoughts on how your project, or how your particular source, and then just your own ideas about it, what these DNA tests imply. I'll click on Ancestry Composition Guide, and this tells you their methods. This company has 45 populations around the globe, and those are the 45 potential places that could show up on your report. How they determine 45, honestly, I'm not entirely sure. Keep in mind what we heard in the podcast about how many different types of people are there out there in the world. They compare your genome, your genetic markers, to there's 14,000 people with known ancestry, and we're going to talk about what that means, known ancestry, in a bit. So those 14,000 people are like the baseline for which they compare every customer to these 14,000. They come from one of 45 populations, and I did a quick calculation here. That means that on average, there's about 311 people, baseline people, for each population. But that's the average. I mean, I can imagine, they don't actually show the sample sizes here, that there's some populations for which they have a lot more people, like 500 or 600 or whatever for baseline. And there's probably some populations for which they only have 100. It's not a great sample size. However, they probably are doing what they can with what they have available to them. Those DNA variants or markers occur in different frequencies in different places. So there could be a particular marker that's really, really common in, say, Europe and parts of West Asia here, and it gets less and less common the farther away you get from that area in purple. So, and they do make this note, and this is important, that they don't do an ancestry profile based on, say, this one DNA marker, which is this one, it's called a haplogroup. They don't sort people just based on this particular piece of DNA that's coded on maybe, I don't know, chromosome 19. That's not the only one they use. They actually have hundreds of thousands of different markers that are spread across all the chromosomes. So they do have quite a few different markers that they use. And then they would only use a particular marker if that marker had a geographic distribution that was obvious when they plotted it on a map, the way that they plotted these frequencies. So they will look at evidence from multiple markers, not just like one or two. It's not like, say, in the podcast when they would just look at, say, shape of your skull. Is it long or is it short? Wrinkle number one. There's lots of wrinkles. People usually have multiple ancestries. And in order to determine this, though, think about think about this kind of circular logic. You have to assume that there is some ideal ancestral population out there and that you can find some people that represent like the perfect example of whatever this ancestral population was. But first, you have to figure out who they are, who qualifies as being, descended directly with very little mixture from elsewhere from this first ancestral population. But what you'll find eventually is that unless you have severe inbreeding, there's always going to be, quote, multiple ancestries. It also depends on how do we define this population up here, even, well, not even genetically, but like, how do we define a particular place or, maybe a cultural history as being a particular population and not maybe either a combination of a couple or perhaps just a subset of another bigger one. I don't know. So this can get kind of circular in terms of usually have multiple ancestries means, well, first of all, we have to define what are these bounded ancestral units? Like this is A, B, C, and D. Oh, we might have thought so, but what if instead B and C are combined and there's another one, E, that we never even thought of, and that A is actually two different ones, A1 and A2. Like, how do we determine what a single ancestral population is versus two or three? So here we're going to kind of get to the meat of defining an ancestral population. And they do have some criteria, like, for instance, they've got to have an area of the world that had a population that apparently was stable, at least as far as we know, before transcontinental travel and migration were common, at least 500 years ago, which hopefully when you see transcontinental travel and migration, we're thinking colonialism. That's when really there were massive population shifts around the planet from continent to continent. So customers are actually the ones who sometimes become the baseline themselves. Not just the DNA, but the research participant tells the company, because apparently you fill out a questionnaire, grandparents. That's their criteria, is that all four of your grandparents are from Finland or whatever the case may be. Then they're like, well, this person is a candidate for, including in our baseline over here. Here we go, confronting bias. And they note that disproportionately focused participants of European descent as the baseline. I should point that out. Not necessarily as the actual customers who submit their DNA, but as their baseline data. And that is because of, yes, it's true, historically biomedical research, a lot of it has taken place in Europe and in the United States, primarily among white populations. So to further discuss this issue of bias, this article on reference population and regions is really interesting. Okay, so in this particular article, they show you the breakdown of populations, so you can get down to those 45. Bigger regional populations are ancestry that goes back several hundred years, so much deeper than the more recent ancestor locations, which are countries. So countries are smaller than regions, according to them, and not surprisingly, countries are going to be much more recent ancestry than bigger areas. That totally makes sense. I do want to pause for just a minute here and think about how are countries defined, aside from biology, just generally. What makes a country a country? What makes one country separate from another country? Something to consider. So they have, broadly speaking, broken down the globe into these 7, well, this category unassigned is not a region, it's just we can't figure out who you are. Which is telling in and of itself. I don't know how often that comes up, but the unassigned. But they have these other 6 larger regions, and then you'll notice that even within those particular sub-regions, I guess, of the larger region of Europe, you have even more fine-grained analysis based on primarily country, not necessarily always, though, Finland. What I want to do is contrast Europe with this particular region, which includes East Asian and Native American. So this region is further broken down into these sub-regions you can see here. And yes, 3 of those sub-regions are in Asia. One of them is in America. And keep in mind, by Native American, we mean North and South America and Mesoamerica. So then notice within, say, one of their sub-regions, like Chinese South, East Asian. There's even more fine-grained analysis by country and by even parts of country. And we get to here to Native American. Um, it's all just like that's it. This one population, a lot of people stretching all the way really to the very tip here of South America, and certainly up here into Northern America. And I would say maybe about here, then we start to see much sparser but still populated area. So I guess my point is that's a huge area, and that is a very diverse area. But they don't have the data. They literally just they don't have the baseline data for those populations for any number of reasons. So it's a pretty broad category, Native American. So just a quick look at Sub-Saharan. We'll talk about this a little bit in lecture as well. Um, Africa is remarkably genetically diverse. If you're going to pick one continent on the planet that has the most genetic diversity within it, it's definitely Sub-Saharan Africa. The rest of the world is just a subset of the variation you find in Africa. So while this seems like it's getting reasonably fine-grained, I'm like, well, yeah, but however, given what we do know about the history and the prehistory of Africa, so culturally, what we know from things like languages, number of languages, and diversity of languages, and also what we know from DNA analyses, really. DNA analyses that aren't necessarily the baselines for 23andMe. What we do know is Africa is remarkably diverse in a lot of different ways, including genetically. So this breakdown is, they're getting better, I would say, than they were 5 years ago or something in terms of baseline populations, but still, they did not have a huge sample size or a lot of precision when it comes to Africa. You kind of get two schools of thought. You get what we call the splitters, and they would say, no, 45 populations isn't enough. We need like 100 populations or more. That you'll get better accuracy, you'll get a better picture of, say, who someone is or where that orchid belongs in the taxonomy of orchids if we just make more and more categories. That's the solution. And then there is the other school of thought, which would be the opposite, and they're usually called the lumpers. And they say, you know what? The problem is you can't go that fine-grained because you'll just endlessly be dividing, and that's pointless and it's silly. Instead, what we should do is just, maybe group by only a few categories. Just lump them all together instead. So they would have maybe, instead of 45, maybe we should only have 10 different regions. And that's more accurate. It's not very precise, but precision is impossible. So these are the two different arguments. And the question is either one of them right?